This study gets at the question of whether tissue-resident immune cells that seed our tissues early in life and may play an important role in protecting us against infections and tumors can be permanently displaced in the context of chronic inflammation. My lab studies the relationship between tissues and immunity, and to do that we're using a human disease called celiac disease that is triggered by gluten. In this disease, gamma delta T cells that are tissue-resident cells are increased not only during the active phase of the disease, but even when gluten is excluded. Why these cells are increased and why they remain increased upon gluten exclusion has always been a big question in the field. And this is the question that Toufic addressed when he joined the lab.
So the primary reason why I joined Bana's lab was because I was very interested in studying human diseases, while also having the capacity to collaborate with a variety of different labs. And during the course of my PhD, I was lucky to collaborate with David Price's lab at Cardiff University, as well as Jamie Rothschild's lab at Monash University, while also having the chance to work very closely with a multitude of physicians here at the University of Chicago. So we were able to get at the question of what is the stability of tissue-resident lymphocytes under chronic inflammatory conditions by studying celiac disease, in particular having the ability to study healthy, active celiac disease patients and those with healed mucosas on a gluten-free diet. In order to address these questions, we took advantage of our ability to get blood samples as well as small intestinal biopsies from patients here at the university and conducted phenotypic, genomic, functional, as well as molecular analyses, specifically focusing on T-cell repertoires to better understand how these cells change over the course of inflammation. During the course of our investigation, we were able to uncover a unique cell type that exists in the healthy mucosa for the duration of life that expressed unique molecules that endowed this cell with the ability to recognize molecules that would allow the immune system to fight and defend against pathogens as well as potential tumors. These T cells are actually dependent on unique molecules expressed by the healthy mucosa called butyrophilin-like molecules. Surprisingly, these butyrophilin molecules were lost in the celiac mucosa, and this loss coincided with the disappearance of this healthy subset of T cells and the emergence of a new cell type with a very specific T cell receptor, which is thought of as a fingerprint of T cells. These cells had the capacity to produce inflammatory cytokines in the context of gluten challenge. Interestingly, although butyrophilin molecules recovered in the mucosa of patients on a gluten-free diet, the healthy immune T cell did not recover, therefore suggesting a permanent reshaping of the tissue-resident compartment.
This has two types of implication. One directly for celiac disease, because it suggests that patients during the active phase of the disease lack cells that can protect them against particular infections and potentially against particular tumors. Which fits the fact that active celiac patients have an increased risk of tumor burden. The second implication is more general, that chronic inflammation can permanently reshape the immune system within tissues, and in particular may eliminate tissue-resident cells that have seeded early in life a tissue and have critical role for tissue homeostasis.
What she said.
What she said.